Dear colleague, my name is Hendro Sudjono Yuwono from Medical Faculty, Universitas Islam Bandung, Indonesia. I want to make a presentation with title Effect of Aqueous Extract of Robusta Coffee compared to neomycin-bacitracin on wound healing by measuring TNF-1 and BFGF in fibroblast cell culture. I will start with my slide. The title of my presentation is Effect of Aqueous Extract of Robusta Coffee compared to neomycin-bacitracin on wound healing by measuring TNF-1 and BFGF in fibroblast cell culture. The co-author is Mirasari Putri, Dr. Mirasari Putri, Dr. Vida Permaningsih, Nora Mufida Marsha, Saset Agustiami Sumantri. The last two is medical students. We are all from Medical Faculty, Universitas Islam Bandung in Bandung, Indonesia. Coffee powder with caffeine in it has an antioxidant, anti-inflammatory, and antimicrobial. So coffee as well promotes cell migration in wound healing. Caffeine itself an adenosine receptor antagonist, so it would negate the effect of adenosine in promoting wound healing. It is by Ojeh in 2016, the title The Effect of Caffeine on Wound Healing. So about the coffee for wound healing, you can see in my publication, international journal, as those. My experiment is in human fibroblast cell culture that takes place at the biomolecular laboratory Areta Medica Utama in Jalan Babagan Jeruk 2 number 9 in Bandung. And coffee powder extract compared to the antibiotic powder, neomycin-bacitracin group, and control in scratch wound healing formation by measuring growth factor B1 transforming growth factor and basic fibroblast growth markers. And in human fibroblast cell culture, it is from ATCC CRL 2522 from USA. Coffee powder extract used is an Arabica coffee powder extract from PTN from Indeso, and neomycin-bacitracin purchased from the Faros Indonesia pharmaceutical industry. This is the picture of cell migration. You see in the quadrant the control and compare with the extract coffee powder of 100 microgram per milliliter. It is very contrast. So that means the best cell migration potential in the formation of scratch wound healing. This is the result of transforming growth factor B1 concentration in coffee. In this picture, we see that the result of concentration of 100 microgram per milliliter is the highest of cell migration. So that means that the concentration is the best cell migration potential in the formation of scratch wound healing. This is the result of coffee BFGF concentration. We see that this is very high compared with the control of the concentration of 100 microgram per milliliter and compared with control. So that means that the BFGF concentration here is very good potential in cell migration. Neomycin-bacitracin migration of cell, we see that the control is high compared with the concentration of 100 microgram per milliliter. That means that at that concentration is the low cell migration potential for scratch wound healing. This is the neomycin-bacitracin result and TGF-beta-1 concentration. That this is in the red arrow. We can see that the concentration is very high. That means it is the best cell migration potential of scratch wound healing. This is the neomycin-bacitracin result of BFGF concentration. We see the red arrow that in concentration of neomycin-bacitracin 10 mg per milliliter is the higher. This is the best migration potential in the formation of scratch wound healing. Based on the test results, it is known that the coffee powder extract at a concentration of 100 microgram per milliliter has the best migration potential for scratch wound healing. And then for neomycin-bacitracin, the concentration of 10 microgram per milliliter is the best, well, for TGF-beta-1 and BFGF. But for migration, cell migration, low potential in concentration of 100 microgram per milliliter. In conclusion, the coffee extract and neomycin-bacitracin had cell migration properties, but coffee gives more robust result in cell migration potential, transforming growth factor beta-1 and BFGF concentration compared to neomycin-bacitracin. Thank you.